Hi everybody, let's go over Watson and Crick DNA. DNA has 4 different nucleotide bases. They are adenine, thymine, cytosine, and guanine. We can think of these as 2 different sets of dance partners. Adenine has been partners with thymine for their whole lives, and cytosine has been partners with guanine for their whole lives. Even though they are each skilled dancers, switching up the partners would result in discomfort and mistakes when they perform. We can think of those mistakes as mutations in DNA. Adenine and guanine are what we call purines because they possess 2 rings, whereas cytosine and thymine are what we call pyrimidines because they possess only 1 ring. Base stacking is simply the organization of these 4 nucleotide bases on the interior of the DNA double helix. They are organized in parallel planes, so adenine is directly parallel to thymine and guanine is directly parallel to cytosine. Going back to the dance partners analogy, adenine and thymine begin opposite in facing one another, as do cytosine and guanine. Base stacking maximizes the hydrophobic interactions between these bases, and these hydrophobic interactions are the main force in stabilizing the double helix. Hydrogen bonds and phosphate backbone are also integral parts to the structure of DNA. Hydrogen bonds are weak and they are not the main force joining the 2 strands of DNA. There are 2 hydrogen bonds between adenine and thymine and 3 between cytosine and guanine. The phosphate backbone is negatively charged and it connects the nucleotides to one another. The backbone is connected through what we call phosphodiester bonds. Here we have a ribonucleic acid. Carbon 1' is attached to the base. Carbon 2' allows us to determine whether the nucleic acid is RNA or DNA because RNA has the oxygen in the 2' carbon and DNA does not. The 3' carbon bonds to the next 5' phosphate to help make the backbone, and the 5' has a phosphate group. There are also what we call major and minor grooves in DNA. These are essentially different parts of DNA where the coil is deeper, wider, and larger for the major groove and smaller or thinner for the minor groove. We also use the terms handedness and antiparallel to describe DNA. Handedness is the direction that the DNA helix coils. Most DNA is right-handed, meaning it coils up and to the right. Antiparallel means that one strand of DNA runs from the 3' to the 5' end, and the other strand runs from the 5' to the 3' end. Now let's look at how salt concentration affects DNA. Tm stands for the midpoint of thermal denaturation. Tm is the point in denaturation where half of the DNA is still double-stranded and the other half has been denatured. In DNA, the phosphate backbones hold a negative charge, and that negative charge is a major reason that denaturation can occur. When both strands are highly negative, they repel each other, and the hydrophobic interactions between the bases are then easier to break. High salt concentrations partially neutralize the negative charges on the backbones, which means they no longer repel each other as strongly. This leads to stronger hydrophobic interactions between the bases, which again is the main force in stabilizing the double helix. The stronger the interactions between the bases, the harder they are to break apart. And the harder they are to break apart, the more energy that they require to do so. In conclusion, the Tm of the DNA in a high salt concentration will be much higher because those bonds are harder to separate and the double helix is more stable. Thank you for listening.